Hey guys, welcome back. This is our last set of notes here for Chapter 6, Photosynthesis. We've talked about our light-dependent and light-independent reactions both so far, and if those weren't confusing enough, we're going to go ahead and jump into some alternative pathways for photosynthesis. And so today we're going to talk about how plants survive in some different environments because there are some hurdles we have to overcome for photosynthesis when we're in a really hot environment or in a really dry environment. So we're going to talk about our C3 plants, our C4 plants, and our CAM plants. And you might recall a little bit of this from general biology. It was from chapter 4, but we'll get right into it here. So we're going to start with stomata. Stomata are a little opening in the plant leaf through that waxy cuticle. And basically they are little pores that allow carbon dioxide into a leaf and oxygen out of a leaf, because you recall oxygen is that waste product of photosynthesis that we want to get out, and carbon dioxide is needed in the Calvin-Benson cycle, so we need to get that in. Stomata are going to close on our dry days to minimize water loss, because this is sort of the dilemma we find ourselves in. I want my stomata open to get carbon dioxide in for photosynthesis, but when they are open, I will lose water out of the stomata. And if I'm in a wet environment, that's really not a big deal. But if I'm in a really dry environment, that can be really costly to lose that water. And sometimes it can almost outweigh the benefit of actually making sugar if you're losing that water. So closing, if we close it, we can save the water, but we don't get the carbon dioxide. And so what we're going to talk about today is how plants overcome that difficulty. So a lot of the problems we get when we close stomata to save that water is a problem that we call photorespiration. You learned in general biology that plants do photosynthesis, where they take carbon dioxide and they take water and they make glucose and they make oxygen, and that humans and animals do respiration, where we take the oxygen and the sugar and we make the carbon dioxide and the water. Now, plants can also undergo respiration. They don't just do photosynthesis, they can do respiration as well. And one particular type of respiration they do is photorespiration. And that happens when we close those stomata to keep that water in, and our oxygen levels get too high because we can't let the oxygen out. And our CO2 levels decline because we're using it all in the Calvin-Benson cycle. And once we get these two different concentrations, and they're so severe that they'll really reduce our efficiency of sugar production at our Calvin-Benson cycle. And so if we get too much oxygen stuck in the leaf, our rubisco, which we need to attach to carbon, will start attaching to oxygen, and it's not going to be a very effective cycle once that starts to happen. So it requires a lot of ATP, and it requires a lot of NADPH, but we still can actually get that to produce sugars. It's just much, much more expensive. So we really want to try to avoid that when we can for plants, because it's going to be very wasteful. And then we're going to end up producing carbon dioxide as opposed to fixing carbon dioxide for sugars. And so that, again, we can make the sugar in the end, but it's really not going to be very efficient. So most of the plants we've talked about so far have been C3 plants. This is probably your most common pathway for fixing carbon and making sugars. These guys use the Calvin-Benson cycle, which we talked about last lecture, to fix carbon, and that's very, very common. And so you know from last time that we form that 3-carbon PGA, and then it turns into PGAL, and then we make some of that into glucose. That's yesterday's stuff, right? That occurs in some mesophyll cells, so cells interior of the leaf that are kind of making up that middle of the leaf. It's used by most of our plants, but it is inefficient in really dry weather. And so if you take most of our plants, our garden and our house plants, and you put them in the desert, they're going to be hurting because they use C3. And C3 works fine in most places in South Dakota, in the Midwest, but it's not going to work fine if it's really hot and dry. And so the example of C3 plants, there are many. They give the example of barley, but really most of the stuff we grow, unless you're growing a cactus in your room for some reason, is all pretty much going to be C3. So here's adaptions for to our photorespiration problem. So here is a little diagram of photorespiration. We have excess carbon, or rather excess oxygen, coming into the Calvin-Benson cycle, and we don't make PGA, we make just PG, we lose some carbon dioxide, and then it takes a lot of ATP and it takes a lot of NADPH to make that PGA. And so we still can, excuse me, we still can make that PGA, and we still can get into that Calvin-Benson cycle, but it takes this long loop around to get there and a lot of extra energy to get there when you guys already know that the Calvin-Benson cycle itself consumes a lot of energy. So we still can make sugars, we're just not efficient if we're doing photorespiration. So let's look at our first way to solve this problem. These guys are called C4 plants. You see it in corn and switchgrass and stuff like that. These guys have an additional set of reactions for sugar production on our really dry days when we have to close stomata, and they're going to compensate for that inefficiency of rubisco trying to bond with oxygen. And so the way they do it is they form a 4-carbon molecule called oxaloacetate. That's where we get C4 and C3, right? C3 was PGA, C4 is oxaloacetate in the mesophyll cells. And then we'll actually move that over to the bundle sheath cell to make our sugar. And so the way we keep photorespiration from occurring is we have 2 separate cells for 2 separate reactions. So we don't get that oxygen buildup from just one cell. So it's kind of a cool trick. Bamboo does it as well. And so here's what it looks like for them. We have mesophyll cells and bundle sheath cells. We get our CO2 from our stomata, and they go through the C4 cycle and makes oxaloacetate. Oxaloacetate moves to a different cell. We get carbon dioxide in this cell as well, and it jumps into the Calvin-Benson cycle. And so we're making sugars, but we're not storing a bunch of excess oxygen in this mesophyll cell. And so we're going to keep that efficiency pretty high in C4. So let's move on to CAM. CAM stands for crassulacean acid metabolism. You guys probably remember these as cacti from Gen Bio, and these guys have an alternative pathway to fix carbon that's going to allow them to conserve water, because not surprisingly, cacti live in environments where it's super dry, and so they need to conserve water. And so these guys are going to form that same 4-carbon oxaloacetate that we saw in C4, but they're going to do it at night, which is cool because they can open their stomata at night and get the carbon dioxide with less water loss via transpiration because it's not super hot. Then in the daytime, they close their stomata because it is super hot. They don't lose water because their stomata are closed, and they will finish photosynthesis in the daytime. And so they've really got this super cool design of separating these two processes to the time of day that they're best suited to, because in a cactus in the desert, if it were to open stomata in the day, it would be dried out super fast. And so that's a really good idea for the CAM plants. And so here's what a CAM plant might look like. We've got night and we've got day. And so night up here is green. They bring in carbon dioxide. They go ahead and make their oxaloacetate, and it just kind of hangs out. In the daytime, we go ahead and move that oxaloacetate over, and we throw them into the Calvin-Benson cycle and make that sugar. And then we repeat the process the next night. So very cool design for tolerating the temperature and the wind in a desert environment. So that is the end of our chapter. I'll leave you with a few points to kind of ponder that we can talk about in class. Let me know what you think. Are greening efforts, such as biofuels and low carbon imprint practices, too little, too late? Why or why not? So I guess the question there would be, is the stuff we're trying to do with efficient vehicles and making biofuels, are we kind of past the point of really trying to incorporate those, or do we need to invest more time and money into those? That's a tough question, I guess. Why is radiation with greater or lesser wavelengths than visible light not used in biological processes? That's a good question. Why is it we only use that little teeny tiny sliver of visible light to power plants? Why can't we use anything else in that giant spectrum of energy that makes it to our planet? And then last question for you to think about: what conflicting needs confront a plant living in a hot, dry environment, and what is the result in a C3 plant if it's in a hot and dry environment, and how do C4 plants avoid that? That third question arguably might be the most important to kind of gauge what you've picked up out of this chapter. So, and I think the answers to that would definitely be in your book and notes and kind of what we talked about today. So I think that's it for this chapter, and thanks for your time.